Hello everyone, my name is Kusafuka Yuko. Let me start my presentation, but first of all, some of you might notice that my research team has changed. My new research team is to measure the plant cell water content using terahertz spectroscopy. So first of all, I want to introduce why I have chosen this theme. One day I thought I want to research about the plant using terahertz technology. However, individual plant itself contains too much water that it is difficult to measure the whole plant because the water is likely to absorb the terahertz radiation. So instead of that, I set plant cell as my sample. The plant cell also contains much water, but because the plant cell size is so small, the absolute amount of water is small too, and it is easier to take information by the terahertz spectroscopy than the plants. So this is the image of the plant cell. The difference between the animal cell is cell wall and chloroplast and vacuole. And cell wall is the cover of the cell made of cellulose, which is likely to transmit the terahertz radiation. And the chloroplast is a material which have a function of photosynthesis. And vacuole is a bag filled with water. In a big mature cell, vacuole could occupy 90% of the whole cell. So the plant cell could be said that it contains much water, and because of this, I think that I can measure the plant cell condition using terahertz spectroscopy because it is easily influenced by the water. Next, I want to discuss about my research objective. Now I am trying to measure the plant cell water content using terahertz spectroscopy. And for the future, I am considering that I could apply this to freshness assessment of the agricultural products because the freshness assessments, because the freshness of the food and the water content has a big relationship with each other. And in my research, I select the onion epidermal tissue as my sample cell. This is the piece of onion cell, and this thin film is the onion epidermal tissue. And the picture below is an image taken by the microscope. And each of these are the plant cell, and this partition is the cell wall, and this little circle is the cell nucleus. And I chose this as my sample because the tissue consists of single layer of plant cell, and the cell size is relatively very large and is easy to observe like this. And also, it is very easy to peel off from the onion. And next, I want to explain about my two recent work, the measurement of the transmittance spectrum and the trial of the measurement of thickness of the sample. First, I will explain about the transmittance measurement. these are the device and the samples I used for the experiment. The sample tissue was peeled off from the second leaf from the external side of the onion, and it is red, and it is the external side tissue is relatively very thin, and it is very likely to transmit the terahertz radiation. Next, these are the experimental methods. First, I peel the onion epidermal tissue into optimum size and stretch and fix it onto the sample holder like this, and place the sample holder into the measurement chamber, which is filled with dry air. And finally, take the transmittance spectrum 4 times with intervals of 20 seconds each. And these are the spectrum which I obtained from the experiment. The blue one is from the first measurement, and 20 seconds after I got the red one, and 20 seconds after I got the green one, and 20 seconds after I got the purple one. And you can see that as the time advances, the transmittance will get higher. So discussions. I consider that this transmittance change is the result of the water content change. The transmittance gets higher because the cell becomes dried. And here, the transmittance have changed about 10% in 1 minute, and it is very speedy. And I think this is because the sample chambers is filled with dried air, and the sample was easily dried up. And from these, sorry. And sorry. From this data, it can be said that it seems to be possible to estimate the water content of the tissue using the terahertz transmittance spectroscopy. And next, the measurement of the thickness. Now I'm trying it. I want the values of the sample thickness because I want to use this to calculate the optical constant and calculate the actual water content of the sample. On the other hand, the thickness of the onion epidermal tissue is said to be about 50 to 120 micrometers when it is fully watered. However, this value will vary from the water content, individuals, and the position of the onion leaf, and so on. But I want to have a precise value of this, so I conduct this measurement. Here, I use a microscope to measure the epidermal tissue thickness because, as I said, the tissue's thickness is about 100 micrometers, and it is thick enough to observe with the microscope. And method is first, I sandwich the epidermal tissue by two cover glasses, and after that, I obtain the image by the microscope from the upper side of the sample. And here, I could obtain an image like this. And by measuring the aperture of the two glasses, I can estimate the thickness of the sample. And these are the results image I got from the microscope. So this blue one, purple one is the cover glasses, and the center one is a sample which are stained with red. And as I said, I want to measure the aperture of these glasses, but these still obtained from the microscope. The image have a problem that the both edges of the cover glasses wouldn't be focused on the same time. And I think this is because the cover glass height has a gap with each other. And now I'm making a trial to make a clear image of both glasses, like layering them like this. And I'm now trying this, so result will be coming soon, I think. So finally, I want to make a summary of my presentation. My research team is to measure the water content of plant cells using terahertz spectroscopy. And until now, it seems that water content seems to be able to measure by the transmittance change of the terahertz radiation. And for next step, I am thinking of measuring the thickness of the epidermal tissue and try other terahertz spectroscopy. That's all. Thank you for listening, and everyone have a happy Halloween next week. Thank you.
Anything? Question?
I know that you want to measure the plant cell, the concentration of the water in plant cells. So, firstly, I don't know what is the goal of your research after you measure the concentration of the water. What is your how do you want to your application? Only last slide, that shows that you want to measure the freshness of the vegetable?
I think it is very good question from you, because I'm thinking it is a big problem about my objective, because I start this research just from my because I was just interested in the plant cell. And now I'm thinking of my application of the research, and now I'm considering that it maybe can apply to freshness assessment of agricultural products, because it has a big relationship with the water content. Is it okay?
I think you can measure another parameter. Concentration of water in vegetable. I think we can go to another parameter. No, I don't say any good solution.
Yeah, I have to think about it much more, I think. Thank you.
You are measuring the water content of the vacuole. Your method assumes that the absorbance of the water is always the same in any vacuole. Is the contents of the vacuole affecting, is there zero effect of the contents of the vacuole on the absorbance of the water in the terahertz region? If the contents have an effect on the absorbance, you will still get a transmission value that is different, but it cannot be quantitatively determined because it changes between cell types.
I didn't think of it. Thank you. now I only measure the onion tissue, so maybe the contents of the vacuole is same, but maybe when I started to measure other cells, I think there will be a difference caused by the content. Thank you.
Anyone else? This is a question. In your microscopic observation, you use the transparent glass cover. Glass covers. And some use the How about using the colored glass cover, for example, black color, or anything like that? So maybe you can easily measure the distance.
Okay, thank you.
This time your sample is the epidermal tissue itself. So the transmission methods may be available. But in a future round, of course, you need to measure the roots or vegetable itself. So maybe you will apply ATR spectroscopy, but in ATR spectroscopy, the penetration depth is only tens of microns, but the cell wall is the same thickness. So inside the water content cannot be seen, maybe in that position in ATR spectroscopy.
セルをセルウォールデプス。セルウォール。
For example, if the cell wall is 10 micrometer and the penetration depth is 20 micrometer, only slight information can be obtained from the ATR spectrum. So the slight change inside the cell may be difficult. So you need to find some other.
Okay, thank you.
Anything else?
Yeah, I think in your study, to know the exact moisture content is important. How do you obtain the actual moisture content from the tissue?
I'm thinking of measuring the
Weight?
weight and volume. Sorry.
Weight and volume is a density.
Density.
You have to get the dry weight of the metal, then the wet weight of the metal, then you can calculate. If you have open to dry.
Thank you.
How about the comparison with the cuticular layer? The most the surface of any plant had the cuticular layer. The cuticular layer consists of some material cutin. That is some kind of wax layer. That is very thin, probably 0.1, 0.05, similar size of the cell. Then the difference is water content. And that's easy to get the cuticular layer thickness or some absorbance. That comparison, just start with that. That might be much difference from cell. I'm sure that that is effective or not in your research, but maybe possible to just compare.
Thank you.
Anyone?
About our application, you want to the refreshness assessment. So I think it's you can talk about the period research, like what the other methods are. So and then you can make some compare and then talk about your research and your disadvantage. I think it's better.
Okay. Thank you very much.
Okay, I think it's time to stop the talking. Alright, yeah, thank you very much.